So through this method, it is possible to detect gains and losses, and also to locate them in the genome. Before giving you the result, I just need to highlight that what we consider important in a gene are different elements. So if this is a string of DNA, what I represented here as rectangles are what is called the coding sequence. So the sequence with ATG and so on that will matter to make the RNA and then make the protein. So this is called the coding sequence. But there are other sequences that are important as well for the gene to function, and these are what are called non-coding sequences that are nevertheless conserved through evolution because they matter, and they matter to make the gene work, function, be expressed, and basically these sequences are necessary to say when and where a gene is expressed in the body, in which cells and at what stage, and how much, of course. And what we found by comparing FSHD cases to control DNAs were several cases carrying deletions of one of these elements. So this is an element that if you delete it, the protein is still capable to be made, but you may interfere with where it's expressed in the body and when. And although we still have to demonstrate what it does exactly, and this is ongoing in the lab, what we wanted to know was how frequently was that mutation associated with FSHD. So we did this screen of whether it was present or not in like a group of 40 healthy individuals, 19 cases of FSHD without contraction, but also a number of cases with FSHD1. And although I don't want to go much in details in the results, but basically on this graph, what you see is in red, the loss, so a case like this, and in blue, all the cases with no loss. So you see that in the healthy population, there's a very small proportion of cases that carry this deletion. So that means by itself, this deletion is not sufficient to cause the disease because it is present in the healthy population. But if you, so this is the sum of all FSHD patients, this plus this, and we had an increase in the representation of that abnormality among FSHD patients. So just to distinguish, we looked at two places, and this place is significantly associated with the cases without contraction. But if we look in another place, we also had some significant association with FSHD1. So that means this is likely to be a genetic modifier. One of these things that if associated with, by itself, it's not sufficient to cause the disease, but if you associate it with either FSHD1 or with a context that is not sufficient by itself to create the disease, then you just start getting the symptoms. And it's extremely interesting to notice that among our 10 FSHD1 cases, we had a couple of twins, both carrying contraction with 3 repeats only, but one of the twins doesn't have the disease and the other one has it. And the one that has the disease was carrying that deletion as well. So although this is not a proof of causality, it's a very remarkable fact that teaches us that we should pursue on that direction. Sorry?
How old are the twins?
I think they are adults.
And
yeah.
Are they fraternal? Are they identical or non-identical?
So they are identical twins, but there are just a few modifications in the genome that are probably somatic mutations that occurred after fertilization. So this genetic defect is not only associated with FSHD, but is predicted to silence the FAT1 gene in some tissues, but not others. And we are currently not only trying to determine in which tissues it's acting, but also if we use it to actually generate a mouse model in which the FAT1 gene is deleted under the control of that element, then we will be able to reproduce, let's say, a very optimal FSHD model. And that's where I want to come with perspective. So basically now we try to use either mice, genetically modified mouse models, but also either cells derived from the mice or cells derived from patients, and including the patients we have identified to carry FAT mutations. And we're going to use them first to develop. So the first thing we can do is, because we have point mutations in some patients, at least for these patients, it is possible to have some individualized therapy. Of course, individualized therapy is a great hope, but it is not applicable to everybody. So we would like to be able to translate this to the next step, which is how can we use our findings to identify therapeutic, like to do some screenings to identify drugs that will be applicable to about everybody. So that's like the main question being how much of FSHD is due to fat deficiency and how much we can try to target this event to try to at least alleviate some of the symptoms. And this is where I will stop and just thank my collaborators again and the fundings, including funding from FSHD Global. Thank you for your attention.